Okay, 15.2 is recombinant DNA. So suppose you have an electronic game that you want to change. Knowing that the game depends on a coded program and a computer microchip, you need a way to get the existing program out of the microchip. Read the program, make the changes you want, and put the modified code back into the microchip. So what does this scenario have to do with genetic engineering? Well, just about everything. So how do scientists copy the DNA of living organisms? Well, the first step in using the polymerase chain reaction method to copy a gene is to heat a piece of DNA, which separates its two strands. Then as the DNA cools, primers bind to the single strands. Next, the DNA polymerase starts copying the region between the primers. These copies can serve as templates to make still more copies. So until recently, plant and animal breeders could only work with variations that already exist in nature, and the changes produced by mutation were unpredictable. But today, genetic engineers can transfer certain genes from one organism to another, designing new living things to meet specific needs. So it's relatively easy to extract DNA from cells and tissues, and the extracted DNA can be cut into fragments of manageable size using restriction enzymes. And these restriction fragments can then be separated according to size using the gel electrophoresis or other similar techniques. There is a picture in your book showing using the extracting of DNA using the gel electrophoresis you need to take a look at. And in finding genes, let's look at a little bit of history here. In 1987, Douglas Preischer, a biologist at Woods Hole Oceanographic Institute in Massachusetts, wanted to find a specific gene in a jellyfish that codes for a molecular green fluorescent protein, or GFP. The natural protein found in the jellyfish absorbs energy from the light and makes part of the jellyfish glow. So Preischer thought that the GFP from the jellyfish could be linked to a protein when it was being made in the cell, a bit like attaching a light bulb to that molecule. So to find the GFP gene, Preischer compared part of the amino acid sequence of the GFP protein to a genetic code table, and he was able to predict a probable messenger RNA-based sequence that would code for this sequence of amino acids. Next, he used a complementary base sequence to attract and a messenger RNA that matched his prediction and would bind to that sequence by base pairing. And then after screening a genetic library with thousands of different messenger RNA sequences from the jellyfish, he found one that bound perfectly. So to find the actual gene that produced GFP, Preischer took a gel in which the restriction fragments from the jellyfish genome had been separated and found that one of the fragments bound tightly to the messenger RNA, and that fragment contained the actual gene for GFP. So this method is called Southern blotting, and that method is actually after the inventor Edwin Southern. Okay, there's a really nice picture in your book on the Southern blot analysis. And let's see. Today, it's often quicker and less expensive for scientists to search for genes in computer databases where the complete genomes of many organisms are available. The polymerase chain reaction. Once biologists find a gene, a technique known as PCR, which means polymerase chain reaction, it allows them to make copies of it. So a piece of DNA is heated, which separates its two strands. And at each end of the original piece of DNA, a biologist adds a short piece of DNA that completes a portion of the sequence. These short pieces are known as primers because they can prepare or prime a place for DNA polymerase to start working. So the DNA polymerase copies the region, between the primers, and these copies then serve as templates to make more copies. And in a way, just a few dozen cycles of replication can produce billions of copies of DNA between the primers. Okay, how is recombinant DNA used? Recombinant DNA technology is joining together DNA from two or more sources. It makes it possible to change the genetic composition of living organisms. Scientists began wondering if it might be possible to change the DNA of a living cell, and many of them realized this had already been accomplished decades earlier in Frederick Griffith's bacterial transformation experiments. And during transformation, a cell takes in DNA from outside the cell, and that added DNA becomes a component of the cell's own genome. Griffin's extract of heat-killed bacteria contained DNA fragments which were taken up by live bacteria, transforming the live bacteria and changing their characteristics. Today, scientists can produce custom-built DNA molecules in the lab and then insert those molecules along with the genes they carry into living cells. Machines known as DNA synthesizers are used to produce short pieces of DNA up to several hundred bases in length. These synthetic sequences can then be joined to natural sequences using DNA ligase or other enzymes that split DNA together. So a gene from one organism can be attached to the DNA of another organism. Restriction enzymes cut DNA at specific sequences, producing sticky ends, which are single-stranded overhangs of DNA. If two DNA molecules are cut with the same restriction enzyme, their sticky ends will bond to a DNA fragment that has the complementary base sequence. DNA ligase then joins the two fragments, so the resulting molecules are called recombinant DNA. So recombinant DNA technology, the joining together DNA from two or more sources, makes it possible to change the genetic composition of living organisms. And by manipulating the DNA in this way, scientists can investigate the structure and function of genes. Scientists working with recombinant DNA soon discovered that many of the DNA molecules that they tried to insert into host cells simply vanished because the cells often did not copy or replicate the added DNA. So today, scientists join recombinant DNA to another piece of DNA containing a replication start signal. This way, whenever the cell copies its own DNA, it copies the recombinant DNA. Okay, next is plasmids and genetic markers. So in addition to their large chromosomes, some bacteria contain small circular DNA molecules known as plasmids. So joining DNA to a plasmid and then using the recombinant plasmid to transform bacteria results in the replication of the newly added DNA along with the rest of the cell's genome. So plasmids used for genetic engineering typically contain a replication start signal called the origin of replication and a restriction enzyme cutting site such as EcoRI. And plasmids are also found in yeast, which are single-celled eukaryotes that can be transformed with recombinant DNA as well. Biologists working with yeast can construct artificial chromosomes containing centromeres, telomeres, and replication start sites. These artificial chromosomes greatly simplify the process of introducing recombinant DNA into the yeast genome. Bacteria can be transformed using recombinant plasmids. Scientists can insert a piece of DNA into a plasmid if both the plasmid and the target DNA have been cut by the same restriction enzymes to create sticky ends. Okay, next, the plasmid DNA transformation using human growth hormone. There's a picture in your book you need to take a look at. Okay, plasmids and genetic markers. The new combination of genes is then returned to a bacterial cell, which replicates the recombinant DNA over and over, and it produces the human growth hormone. The recombinant plasma has a genetic marker, such as a gene for antibiotic resistance. A genetic marker is a gene that makes it possible to distinguish between bacteria that carry the plasmid from those that don't. So this plasmid contains the antibiotic resistant genes, and after transformation, the bacteria culture is treated with an antibiotic, and only those cells that have been transformed survive, because only they carry the resistance gene. So how can genes from one organism be inserted into another organism? Well, transgenic organisms can be produced by the insertion of recombinant DNA into the genome of a host organism. The universal nature of the genetic code makes it possible to conduct organisms that are transgenic, containing genes from other species. Transgenic organisms can be produced by the insertion of recombinant DNA into the genome of a host organism. Like bacterial plasmids, the DNA molecules used for transformation of plant and animal cells contain genetic markers that help scientists identify which cells have been transformed. Transgenic technology was perfected using mice in the 1980s, and genetic engineers can now produce transgenic plants, animals, and microorganisms. By examining the traits of a genetically modified organism, it is possible to learn about the function of the transferred gene. So many plant cells are transformed using agrobacterium. In nature, this bacterium inserts a small DNA plasmid that produces tumors in a plant's cells. Scientists can deactivate the plasmid's tumor-producing gene and replace it with a piece of recombinant DNA. The recombinant plasma can then be used to infect and transform plant cells. The transformed cells can be cultured to produce adult plants. Transgenic plants transform